In this video, we're going to be taking a look at three topics, 1.3 through 1.5, Introduction to Macromolecules, Carbohydrates, and Lipids. The organic carbon-based molecules that are important to the function of living things can be classified based on their structure and function. The four most important of these organic molecules are carbohydrates, lipids, nucleic acids, and proteins. Three of the four, carbohydrates, nucleic acids, and proteins, are considered polymers. This is due to the fact that the individual building blocks, or monomers, of those macromolecules all have the same basic structure. The suffix "mer" means unit or piece, and the prefix mono denotes one, whereas poly means many. Lipids, on the other hand, are constructed of different kinds of building blocks, which we'll see later in this presentation. Each of those groups of macromolecules has responsibilities in the sustainment and propagation of life. Regardless of the category of macromolecule in question, all are constructed using the same chemical process. This process is called dehydration synthesis. The reason for this name is because it involves the removal of atoms from the smaller reactant molecules in order to form water and build a larger product molecule in the process. If you recall from a previous video that functional groups are the collections of atoms at which chemical reactions in organic molecules occur, you'll easily recognize the hydroxyl group participating in dehydration synthesis. We will see specific examples of this reaction when we explore the individual macromolecules one by one. The process used to deconstruct macromolecules is simply the reverse of dehydration synthesis. Hydrolysis requires water as a reactant, as it is necessary to separate the individual building blocks, monomers, from the larger polymer. Hydrolysis uses the deconstruction of water, H2O, to restore the hydroxyl group, thereby breaking the covalent bond holding the polymer together. The hydrolysis of macromolecules is one of the early steps in the digestion of foods. Both dehydration synthesis and hydrolysis are reactions that are catalyzed by a variety of enzymes, which we'll learn more about in a later unit. The first macromolecule that we're going to take a look at is carbohydrates. Carbohydrates, or carbs, are also known as sugars and are the primary source of energy for practically all cells. Some common examples of the simplest carbohydrates are glucose, fructose, and galactose. These are known as hexose sugars because they each possess 6 carbon atoms in their structure. Deoxyribose and ribose, found in DNA and RNA respectively, are examples of pentose sugars, as they contain 5 carbon atoms each. Those individual monomers are called monosaccharides and are commonly linked together to form more complex carbohydrates. This is accomplished via dehydration synthesis and forms a bond between two monosaccharides called a glycosidic linkage. These more complex carbohydrates are important in longer-term storage of energy and in the formation of some organisms' structural components. Plants, for example, store carbohydrates in both chain-like structures and branched structures, collectively referred to as starch. Animals, on the other hand, store carbohydrates in the form of glycogen, a highly branched, complex carbohydrate. In the exoskeletons of arthropods, like insects, spiders, and crustaceans, as well as the cell walls of fungi, bundles of carbohydrate chains called chitin provide for structure, strength, and protection. The most abundant organic molecule on Earth is a carbohydrate. This carbohydrate, called cellulose, is used to construct the cell walls of plants. In nutritional terms, cellulose is the important part of a healthy diet called fiber. It is comprised of long chains of glucose molecules stacked on top of one another, locked in place by hydrogen bonds. This arrangement confers great strength to the plant cell walls individually, and summed up for the entire organism, makes it possible for plants to grow to great sizes. Now, let's see a bit about lipids. Lipids are an important energy source molecule necessary for the formation of cell membranes and help to provide cushioning and insulation for organisms. Lipids are also known as fats, and unlike carbs, proteins, and nucleic acids, are not considered to be polymers. This is due to the fact that two very different kinds of subunits are required to build them. Constructing a lipid requires a glycerol molecule and fatty acids. Glycerol is a 3-carbon compound with 3 hydroxyl groups. Fatty acids are long hydrocarbon chains with a carboxyl group found at one end. Fatty acids can vary in their length from only 4 carbons, like the butyric acid found in butter, to the very long fatty acid chains with 22 or more carbons. Although lipids are not polymers, they are constructed using the same dehydration synthesis reaction. The bond formed between a fatty acid and a glycerol is called an ester linkage. When 3 fatty acids are linked to a single glycerol, a triglyceride is the result. All glycerol molecules are identical, but the same cannot be said of fatty acids. As mentioned earlier, fatty acids differ in their length based on the number of carbon atoms present, as well as the types of covalent bonds found in the chain. Fatty acid chains that are comprised solely of single covalent bonds are called saturated. On the other hand, the presence of a double covalent bond results in an unsaturated fat. Polyunsaturated fatty acids contain more than one double covalent bond. Saturated and unsaturated fats have different chemical properties because of their bonding structure. Saturated fats tend to be solid at room temperature and are more prevalent in many animal tissues. As a part of our diet, saturated fats have more deleterious health consequences. They are more easily stored by cells thanks to their flat linear structure, and result in an increase in low-density lipoprotein, or LDL cholesterol, which is considered to be the bad cholesterol. Unsaturated fats tend to be liquid at room temperature and are more commonly found in plant tissue. Because of the bend or kink in the carbon acid chain caused by the double bond present, they're not as easily stored in cells and cause high-density lipoprotein, HDL cholesterol, the good cholesterol, to rise. The proportion of saturated and unsaturated fats found in the lipids that make cell membranes influence the membrane's fluidity. We'll learn more about the significance of that in Unit 2. A steroid is a type of lipid molecule characterized by a distinctive structure composed of 4 interconnected carbon rings. The core structure is known as the steroid nucleus. Despite this shared backbone, different steroids have varying functional groups and side chains attached to those rings, which give each steroid its unique chemical properties and biological functions. The arrangement of atoms in these rings, including the position and orientation of the bonds between them, contributes to the molecule's 3-dimensional shape. Small structural changes, such as the addition of hydroxyl groups, methyl groups, or double bonds, can dramatically alter the activity and role of a steroid in the body. Since steroids are nonpolar and hydrophobic, they can easily pass through the lipid bilayer of cell membranes. This property allows steroid hormones to enter cells and influence cellular activity. One important hormone is cortisol, a glucocorticoid produced by the adrenal cortex. Cortisol helps regulate metabolism, suppress inflammation, and manage the body's response to stress. Another example is aldosterone, which plays a key role in maintaining blood pressure and fluid balance by acting on the kidneys. It does so by helping the body retain water and stabilize blood volume and blood pressure. Sex hormones, like testosterone and progesterone, play a role in the development of secondary sex characteristics during puberty and the reproductive cycle. Cholesterol has a characteristic steroid structure made up of 4 fused hydrocarbon rings. Attached to this ring are several important functional groups, including a hydroxyl group, which gives cholesterol a slight polarity and allows it to interact with the polar heads of phospholipids in cell membranes, as well as methyl groups and hydrocarbon tails, which are nonpolar, allowing that part of the molecule to interact with the nonpolar portion of the cell membrane. Because of its amphiphilic structure, having both a polar and nonpolar region, cholesterol can insert itself between the phospholipids in a membrane. Its rigid rings help to maintain membrane stability and flexibility across temperature changes. This structural characteristic of the cell membrane, as well as the consequences to its function, will be explored in greater detail in Unit 2. A special and critically important kind of lipid is called a phospholipid. While all other lipids are hydrophobic, phospholipids are amphiphilic. This means that the molecule possesses both hydrophilic and hydrophobic regions. The fatty acid tails bonded to the glycerol are unable to interact with water due to their nonpolar nature. The hydrophilic head region is comprised of a phosphate group and choline, both of which have charges, allowing the head to interact readily with water. When 2 layers of phospholipids are used to construct a cell's membrane, the hydrophilic head is in contact with the aqueous internal environment of the cell, as well as the aqueous external environment. The central region of the membrane, where the tails are found, are oriented away from water, establishing a hydrophobic core of the membrane that is largely water-free. It is this phospholipid bilayer, along with a number of other molecules, that forms the boundary between the external environment and the inside of the cell with its contents. A more detailed look at the structure and function of a cell's membrane will be presented in Unit 2. That concludes the video. Thanks for watching, and until next time, take care.